Very warm welcome to one and all. So, a very good morning to Professor Philipp Simon, and good evening to all the respected scientists and students of UHS Bagalkot and different universities. So, this lecture is organized on behalf of NAHP-IDP. I thank Professor Philipp Simon for accepting my request for giving his experience, sharing his experience with our scientists and students. And I would like to introduce about the today's speaker, Professor Philipp Simon. So I'm very honored to introduce about him. So Professor Philipp Simon is a USDA-ARS research geneticist, a pioneer plant breeder, and a professor of horticulture at the University of Wisconsin, Madison, USA. He has he has contributed significantly in carrot in terms of breeding, genetics, genomics. He targeted important traits like improved flavor, nutritional quality, root shape, nematode resistance, alternaria leaf blight, cavity spot disease, and many biotic and abiotic stress resistance breeding. He led the development with high carotene content, sweet or mild flavor, purple color carrots as well. He has identified and developed elite inbreed lines for disease resistance, male sterility, and many more. He is having more than 700 plant introductions with him, and also a number of germplasm lines. So to complement his breeding effort with students and collaborators, he has developed various breeding tools, including the high-quality genome sequence of carrot in 2016 and the version 2 going to be released shortly. So his team is also has initiated one more big project, Carrotomics database, a carrot genetic genomic and breeding resource that is very much helpful for all the carrot researchers across the globe. They have resequenced important plant introductions and the number of germplasm lines, and they are working on genomic selections, gene editing, and many other concepts. He has also contributed in terms of 10 germplasm collecting expeditions in carrot, onion, garlic, cucumber, and other vegetables. He is a pioneer in producing true seeds in garlic, and also developed orange flesh cucumber with elevated carotene. Till now, he has supervised more than 50 PhD and 15 postdoctorate students from different countries. He has more than 250 research publications to his credit. In the last 7 to 8 years after sequencing of the genome, he has mobilized worth of more than $50 million project grants as a project director. He has research collaborations with Poland, Turkey, Argentina, Bangladesh, Pakistan, Brazil, and to some extent in India. He has been recognized with a number of awards and achievements. A few of them are like he is a research leader of USDA-ARS Vegetable Research Unit, and he served as a US plant breeding coordinating chair. He has been honored with the honorary doctorate from Poland Krakow University, and a fellow American Society for Horticulture Science in 2002, and the Senior Scientist of the Year from USDA-ARS. So, many other awards to his credit. He has worked with a great scientists like Professor Pelaquin, a great cytogeneticist and a geneticist, who hosted Professor Swaminathan as a short-term visiting scientist during the 1960s. He also worked with other scientists like Clint Peterson, MS male sterility in carrot and hybrid development. So overall, he has revolutionized US carrot breeding in the last 50 years. Apart from his contribution, he is the most respected and a generous plant breeder, a global leader in carrot breeding, and a great human being who supports, encourages scientists across the globe, including me, in terms of knowledge sharing or germplasm lines sharing. Truly, he is my inspiration. He has been associated with carrot growers, seed industries across the globe. He has hosted me for a month at University of Wisconsin, Madison, that sponsored under NAHP-IDP project of UHS Bagalkot. We are also looking forward for having MOU, germplasm exchange, and a few collaborative research grants with Professor Simon. And with this introduction, I request Professor Philipp Simon, sir, to share his research experience on building better carrots. So I once again welcome one and all for the event. Professor Simon, our Honorable Vice Chancellor, Indiresh, Dr. K. Indiresh is also present here, sir. And our professor, our Director of Research, Professor Maheshwarappa sir, and other key officers are also present with us.
Okay. Well, thank you for that generous introduction.
Thank you.
Have you any other announcements, Dr. Cholan?
Sir, Professor, our Honorable Vice Chancellor, Dr. K. Indiresh, Director of Research, Dr. Maheshwarappa sir, also have joined this lecture, sir.
Oh, thank you.
Yeah.
Is my
good morning, sir.
Good morning. Good evening. Good morning to you, Dr. Cholan, and good evening to the rest of you. I appreciate your time and interest in hearing about carrots today. And thank you very much for that generous introduction, Dr. Cholan. It has been a pleasure hosting you as a visitor here in the last better part of a month, and look forward to some continuing interactions at several different levels as we move forward with things. Let's see, can I start sharing my slides here? Can you see those? Can you see the slides? Not yet. Okay, let's see. Do I?
Vanilla?
Not yet.
You made it enabled, huh?
Given permission to.
Okay. I think she has given.
Okay.
Okay.
Here we go. Let's see how this goes.
Yeah, it's opening.
Yeah.
Yes. So just slide share.
Right. How's that?
And that's fine, sir. Now.
Yeah, fine. Yeah.
Okay. Excellent. Thank you. Well, again, thank you very much for participating in listening to my discussion of carrots. And again, I really appreciate my opportunities to be interacting with Dr. Cholan and hopefully with others of you as well. I'm going to primarily talk about the work we've been doing for carrot breeding, looking at what we've been focusing on improving carrot for both the carrot growers and consumers in the US, but with some global application as well. Just to give a very brief background, a carrot is, of course, a vegetable, and there's somewhere between 200 and 300 different vegetables. For some vegetables, there's not much breeding, but for about 35 vegetables, there's some fairly extensive breeding. And vegetables like fruits and other horticultural crops have high economic value, but also high resource input for their production. And they occupy a relatively low amount of production land compared to staple crops. In the US in 2020, carrots were worth about $840 million to US farmers. And that puts carrots in the middle of the top 10 vegetables, both in the US and globally. Carrots are an important source of nutrients in the diet, and I'll be speaking more about this as I go along. Is my volume and visuals okay, Dr. Cholan?
They're clear, sir.
Okay.
Clear.
Okay, good. Thank you. Just want to make sure everything is on target. So a very brief overview of where the carrot crop is today, globally speaking. Most carrots grown globally are orange. You in India are very much more familiar with red carrots, which are something relatively new in the US, but I know you grow orange carrots as well. In Asia, the predominant root shape is the Kuroda shape, shown on the far left here, whereas in most of the rest of the world, it's the Nantes shape, which is sort of a cylindrical carrot shown next to the Kuroda. In the US and all of North America, we have a different shape of carrots that are typically grown, long, thinner carrots known as imperator types. And so these are the major shapes of carrots, and again, orange is the major color. One trend globally for carrots in the US, and to some extent more extensively in the rest of the world, there's more consumption of carrots as a fresh crop as opposed to processed or cooked. If we look at this trend line of global per capita carrot availability, so this is the amount of carrots produced and available to each of us globally on the average. the per capita availability has increased in the last 50 years. Again, carrots in the roughly middle of the top 10 vegetable crops and worth 800 million and a source of vitamin A. So in spite of ongoing challenges for carrot growers, the production of carrot has increased over the last 50 years. We hope we can continue this, and I'll talk to you about some approaches to that. Some of the variables that affect the production of carrots today relate to the nature of the ability to grow the crop. Carrot's an interesting crop because some of the same cultivars or varieties can be grown at the Arctic Circle in the summertime and at the equator in the winter. The value of the primary root crop is stable or rising globally. In the US and hopefully globally, there is an increased production of carrots with organic production. It's a crop that doesn't require quite as much pesticides and insecticides as other crops. About 15% of the US carrots grown are grown organically. And there's some new high value crops, including these baby carrots shown to the right in this picture, where these are long, thin carrots, about 1 to 2 centimeters in diameter, cut into pieces and peeled and sold as fresh carrots very widely, over half the production in the US. And this is a high value crop. The value of the seed crop of carrots is relatively low, and because of this, there's not as much interest in carrot breeding for seed companies, but there are some fairly extensive carrot breeding efforts in seed companies. Like most crops, there are new challenges, and like all crops, there are more challenges for adaptation for some of the climate change variables like heat, drought, and salinity that are facing the entire world. If we look at what the grower and the consumer of carrots are looking for, if you talk to growers, they're looking for a very uniform crop. And this was really the big impetus for the development of hybrid carrots in the 1950s and 60s. I'll talk more about in a minute. Of course, growers would like to have a crop they can grow free of diseases and pests and other defects that can limit the sale of carrots. Something like nematode resistance shown in the carrot to the left below is a good example of nematode resistance that has a great deal of interest for growers. From a consumer standpoint, as I said before, fresh carrots or raw carrots are becoming more popular, and from that perspective, sweet flavor and a non-harsh or non-turpentine flavor is desirable with a succulent texture. For most of the U.S. crop, orange color is preferred, although we are developing other colors of carrots. And for the U.S., these baby carrots, again shown on this picture, is impetus for selling carrots that are more convenient for consumers to eat. You just open up the bag and eat them. You don't need to cut them and peel them, which in my opinion isn't a big effort, but to a lot of U.S. consumers it is. And consumers realize carrots are a nutritious crop, and so we've focused on this as well. Although nutritional quality has no economic value for farmers, we still value it for consumers. So a very brief overview of where we get nutrients from plant sources in the diet in the US. This table is very simplistic and somewhat out of date, but still very pertinent today. If we look at the major nutrients that we consume that come from plants in the US diet, carbohydrates, proteins, and fats are significantly represented, but primarily from cereal crops for carbohydrates and protein, where 4 asterisks here mean that more than half of the carbohydrates are coming from cereal crops here, shown in this slide. So these are some of the macronutrients, and again, that's what the staple crops provide for us primarily. The staple crops to the far right of this slide do provide some vitamin E, magnesium, and fiber, showing some micronutrients, just a subset of them below. But most of the micronutrients in the U.S. diet that come from plant sources don't come from the staple crops, but rather from horticultural crops, fruits and vegetables and potatoes. Vitamin A is a particularly high a source of more than 20% of the U.S. Vitamin A comes directly from plant sources that we eat, as opposed to animal sources. And I'll talk more about that as we go along. Won't be surprised that most of the vitamin C in the U.S. diet comes from fruits, citrus in particular, but other fruits and vegetables as well. Potassium, vitamin E, and magnesium are also important micronutrients that come from plant sources. Now, one thing I'll note on the left is the red highlighting, and these denote nutrients among this short list, certainly not complete. These micronutrients that are highlighted in red are shortfall nutrients, meaning that U.S. consumers generally eat enough foods to get these nutrients, but some populations in the U.S. are borderline approaching deficiency. We see no overt deficiency, but consumers in the U.S. should be eating more of these micronutrients. And interestingly, all of those shown here come from vegetables and fruits. So that means we should be eating more fruits and vegetables. And in fact, if we look at what nutritionists recommend in the U.S., less than 40%, in fact, probably less than 35% of the U.S. population eats the recommended 3 servings of vegetables a day. And this shortfall in vegetable consumption is important. The Center for Disease Control has focused on, because it will reduce the healthcare costs for the U.S. U.S. has some significant levels of obesity, heart disease, stroke, diabetes, and cancer, and most of these diseases can be at least alleviated if we in the U.S. would be eating more vegetables and fruits. So from a carrot context, I'll be talking to you about that today. And as a carrot breeder, I asked the question, what can I do to breed carrots to contribute to both crop production and consumption of the crop? And can genetic diversity in the carrot germplasm be available? Is it there to help improve carrots? Because genetic diversity in crop germplasms, or for that matter, domesticated animal germplasm, is the engine that genetic diversity is the engine that drives carrot breeding and the genetic improvement of other crops. So as a crop breeder, that's the primary question I focus on a day-to-day basis. And I'm sure several of you in the audience do as well for your crops you focus on. So if we're looking at this question of use of germplasm, we need to have good collections of germplasm and we need to evaluate it. And so we've had some extensive efforts in evaluating some field traits in some fairly large projects for carrots in recent years. We've looked at consumer traits and also have been developing molecular markers for carrots. I'll talk about all these in more detail as we go along. But before I talk about those in more detail, let's talk about the genetic diversity of carrots. And I think that paying some attention to the process of domestication of carrots gives us some insights into both the origins of carrot, the diversity of carrots, and some insights where we can go in the future to continue to improve carrots. So carrots originated from a wild version of the crop, which is known as Queen Anne's lace, that grows very widely in the U.S. and over much of the world. I believe there's some wild carrot in northern India, and there certainly is in the neighboring countries to your east, to your west. So wild carrot does occur widely in the world. The 2 photos on the left show wild carrot in bloom in a vacant lot in Wisconsin down the road from where I live. And on the lower picture on the left is some wild carrot. You can't see it's really carrot, but that entire small field is a vacant lot in a village in Tunisia. So it's a volunteer wild plant that grows widely. And the crops of it don't really look a lot like carrot. It is a root crop, even as a wild crop, but the roots are very heavily divided and white in color. It's called Queen Anne's lace because of the shape of the flower shown on the photos on the bottom of this. I'll present in the next couple of slides. We know that the origin of wild carrot and carrots globally because of it came from your part of the world in Asia, in particular Central Asia, and the region for you in India and to your west over to Central or Eastern Europe. There are about 20 species of Daucus, Carotis Daucus carota. There are about 20 species of Daucus. Most of those species are in the Mediterranean region, but carrot is unique among the Daucus species in originating as a crop in Asia. I'm not going to go through this in detail, but we've to ask the question of where carrots came from, where cultivated carrots came from. Most of this phylogenetic tree shown here on the right in blue is cultivated carrots, showing the genetic relationship of those cultivated carrots with wild carrots. You see wild carrots in Central Asia being on the middle bottom of this slide, indicating that they're most closely related to cultivated relative to wild carrots in Greece, in Europe, in North Africa, or in the US. So this is molecular genetic information indicating where wild carrots originated from. In fact, the rather small amount of written history also indicates that's where carrots originated in Central Asia. Our molecular study was based on evaluating 88 wild and cultivated carrots to get a molecular fingerprint of them and to evaluate them to, again, ask this question, where did this crop come from? This does agree with the historical record that carrots probably originated only about 1,100 years ago, so relatively recently compared to many crops like wheat and barley, for instance, or rice. Only 1,100 years ago, roughly, based on some written history. Vavilov thought carrots probably originated in Central Asia, and there's some possible records of carrots even in the Roman Empire, but those are not validated. So based on both the scant written record of historical records and the molecular diversity analysis, we've concluded carrots did, as a crop, originated from Central Asia. That's important when we think about collecting wild crop germplasm, because that's where the greatest genetic diversity of the crop, in fact, is today. And so one thing I will note, as I said earlier, sometimes looking at domestication can give us some insights in where we might be going with the crop in the future. Carrots usually referred to, at least in the US, as a cool season crop, but in fact, it originated in a hot, dry environment. This is some relatively good news to us as carrot geneticists and breeders, because this indicates that carrot might have some genetic variation that gives some tolerance to these more abiotically stressful conditions that are confronting carrot growers, and for that matter, all crop growers today. So looking a little bit more at carrot prehistory there, while wild carrot originated in Central Asia, it spread all over Europe at least, and probably all over Asia as long ago as 3,000 to 5,000 years ago, based on finding wild carrot seeds, shown here on the left, in campsites in Germany and Switzerland by some archaeologists. This is probably, these were in campsites giving evidence of first human use, probably quite certainly not using wild carrot roots, although they are edible when they're not flowering, but probably using carrots, these carrot seeds as a spice, like the other members of the APAC, which include crops like caraway, fennel, coriander that we're all familiar with as spice types of crops. And again, these carrots that were in these campsites of our ancient human relatives several thousand years ago probably were collected from wild plants and quite certainly not cultivated. So carrot has been occurring as a weed worldwide, or quite widely, probably not in North America, but for thousands of years, but only as a root crop with certainty about 1,100 years ago in the area of Central Asia, maybe Afghanistan, but not certainly. The first carrots colors were actually purple and yellow, not orange. And I mentioned before some evidence of possible carrots in the Roman Empire, but not well documented. Carrots spread to the east and to the west with probably traders moving around selling their goods. For instance, the European Marco Polo traveling to China probably was carrying carrot seed along the route one way or the other. The first orange carrots were actually relatively recent, only about 500 years ago. And I'll talk a little bit about the development of hybrid carrots, and that's really very recent, of course, only since the 1950s. I'll mention very briefly that the color of carrots isn't so much based on written history. We've had some very smart research efforts going to art galleries and looking at the colors of carrots over time. And based on that, we have a pretty good idea, at least for the Western carrots that are grown, of the color of them. And again, purple and yellow being the first colors, first orange carrots showing up in Southern Europe in the 1500s. And carrots spread throughout the world pretty much since they began, but now they're certainly grown globally. And with this diversity that we see in starting with wild carrots, and there's one wild carrot in the bottom of this photo on the left, with this diversity, we see a wide range of shapes of carrot roots, some indication that there's a lot of genetic diversity of this crop to complement the observations we've made with more molecular genetic approaches. And this variation in the carrot genome is the raw material that gave early farmers and also modern breeders something to work with to improve the crop, to drive domestication and to drive current improvement from this wild crop on the left to the cultivated versions of the crop shown on the right of this slide. So this germplasm of carrot in germplasm collections are a very valuable source for us carrot breeders. And we've tapped into some of that diversity, looking at about half of the carrot germplasm in the US collection, which includes about 1,500 different carrots from around the world. About half are cultivated, about half are wild. And we've done a fairly extensive evaluation of the cultivated part of that collection. We're starting to look at the wild carrots more extensively. The UK also has a fairly large collection of carrots that are available. One thing I'll point out is if you're interested in germplasm of carrots or any other crop, you can get it just for asking for it at ars-grin.gov, where you can find the collection of plant germplasm, and they will send you seed free of charge if you're interested in looking at diversity in the crop that you work, the diversity of the carrot of the carrot crop. When we published the carrot genome in 2016, we only looked at the genetic and molecular diversity in 8 wild carrots and 18 cultivated carrots. And as Dr. Cholin mentioned, we've more recently got a larger database called Carrotomics. When we looked at this diversity in the sequencing of the carrot genome, we found diversity over the 9 chromosomes of carrots, and and some chromosomes shown above and to the left, and then these circles to the right indicate the diversity in those 8 wild and 18 cultivated carrots, shown each as a circle for each to the right in this ring of circles. So we saw quite a bit of diversity in the crop, and this diversity then is what we're working with in terms of breeding, and what we're tracking with molecular markers to improve the crop with some of the tools that are being developed today, again, because this diversity is so important. So let's look at how we're using the diversity and using some of these molecular tools to improve the crop. I will note that carrots are a relatively slow crop to breed because we handle, and most carrot breeders handle, carrots on an annual basis. Once a year is good, but it'd be nice if we could grow the seed for a carrot crop to have a cycle shorter than a full year, like some crops, like beans, for instance, But for carrots, we have to, at this point, live with the fact that we have a 1-year cycle to improve the crop as we're improving it in the breeding program. Another point to note for carrot breeding, and a reason that I'm really glad to be working with Dr. Cholin, is that there are very few of us looking at carrot breeding programs globally. Science works a lot better if you've got people to collaborate with, and I'm looking forward to collaborate with Dr. Cholin and perhaps with others of you. But so these are some background points of interest for looking at where we're at in carrot breeding. And the first focus on carrot hybrids was originated in the 1960s. I'll talk more about this, but this gives you some numbers of the programs that have been breeding carrots, about 5 in Europe, several in Asia. There historically were 12 public programs in North America. Right now, we're down to only 2 public programs in North America, as I point out below on this slide. There were about 25 seed companies working on carrots in the 1960s. That's down to about a dozen today. But those 12 or so seed companies working on carrots are large, and they have very well-developed efforts looking at carrot improvement. Again, primarily focusing on hybrids, but to some extent open pollinated carrots, cultivars. A couple of background points of information for those of you in the audience that are breeders. Carrots, a diploid outcrossing crop that's pollinated by insects, has 18 chromosomes, has a relatively small genome, about the same size as rice, which when I started working on carrots wasn't much of an important fact, but it turns out that as we have started using molecular tools, that's very important because we can make advances more quickly with a small genome crop. There's no self-incompatibility, but there is quite a bit of inbreeding depression. Again, I'll just mention that in passing without saying more, but for those of you breeders, it's of interest. There is a wild and cultivated diverse germplasm. Carrots referred to as a biennial, which historically it probably was in Northern Europe. I don't think it was in your part of the world. I think you've handled carrots as an annual since its origins. But for the Western carrots in Europe and the US, it's handled as an annual, but it's referred to as a biennial because of the selection for vernalization in the crop in Western Europe and in North America. In the 1980s, there were only 20 trait genes known for carrot, and none were on any genetic map. Today, we've got about 200 trait genes mapped, and we've got some heritability measures for about 25. We sequenced the genome in 2016, and this is giving us an abundance of genetic markers which really facilitate and speed up the breeding process. I'll mention carrot pollinations are very difficult, and I'll show you why in the next couple of slides, not quite yet. Looking at hybrid carrot breeding, it's a phenotypic recurrent selection program, meaning that we look at carrots and evaluate them every year and select the best ones and intercross them. Most of the new cultivars are hybrids, a few open pollinated cultivars. The breeding process for developing hybrid carrots starts with self-pollinating the parental stocks for 4 to 6 generations, starting with backcrossing to sterile cytoplasm. I'll show you in the next slide at the F3 level, and then breeding out to about the F4 to F5 to develop inbreds to make hybrids for the crop. This is what we use for the initial evaluation of the carrot crop, is grow 1 meter plots in the field. We invite growers and seed companies and researchers in to judge the crop. And it's basically a beauty contest, but it works very well to give some initial evaluations, finding what we see as a crop, carrot shown on the left, an experimental hybrid from my program used for making baby carrots, these short pieces of carrots, and to the right, the full-length carrots that are sold in plastic bags in the US. And a big carrot field there. On the upper right's a carrot harvesting device used by the big growers. They harvest about 1 ton of carrots in 10 to 15 minutes with these carrot harvesters. So it's like an Orange River coming off of that device, that harvesting machine. And so it's a quite efficiently grown crop on a large scale. primarily in California. Once we harvest the crop, we do some visual selection. Some of the traits here I'll talk about some more as we go along. Internal color, the shape of the carrots, the shape of the shoulders, and also a little picture here showing hybrid vigor between two inbreds of the crop. But we involve quite a few people when we undertake these selection efforts in carrots, because quite a bit of it is visual, and then also tasting carrots as we go along. As we find carrots to produce seed on, we produce seed from individual plants or larger groups of plants in pollinating cages like these, starting with the upper part of the carrot root shown in the lower left part of the image. And then with the carrot flowers, just to talk about the pollination process for carrots, each umbel, which can be quite large. Dr. Cholin tells me that your umbels of carrots in India are larger than ours here. Ours are probably not much larger than or smaller than a soccer ball in diameter, probably quite more like a softball. But so each umbel includes thousands of flowers, and because of this, you can understand where doing hand pollinations would be very, very difficult, and you only get a maximum of 2 seeds for any one flower, but many seeds for the inflorescence. In order to develop hybrid carrots, a source of male sterile carrots, or carrots that do not produce pollen, had to be developed. And the way the normal carrot flowers look is in image A here, where you have petals on the outside, anthers and stamens in the middle sticking out, 5 of them, and then in the middle, the pistil that can house up to 2 seeds. For the male sterile carrots, there are types that have no pollen, shown in image B, and another interesting type that's widely used where where the anthers are replaced by another round of petals, making them fully male sterile, but they are female fertile. So these were important observations made in the 1950s that drove the development of hybrid carrots. Interestingly, this carrot shown in image C, this type of flower only shows up relatively rarely in wild carrots, and it was initially discovered in the 1950s by a researcher at Cornell University. In the 1950s, 60s, and 70s, my predecessor, Clint Peterson, shown in the upper picture, and Warren Gobelman, who was a professor here in the horticulture department in Wisconsin. These 2 individuals were the 2 that initially developed the first hybrid carrots using this source of male sterility that I showed you. And so they were really the real innovators for carrots in the 50s and 60s that got the hybrid development going. So carrot seed production is done in large fields, primarily in eastern Washington state and eastern Oregon and Idaho in the U.S., other parts of the world as well, but for the U.S. that's where seed is grown, pollinated by bees. So looking at breeding goals for carrots today, we're looking at improving the crop for growers and improving the crop for consumers. Again, I showed you this picture before. From a grower standpoint, appearance is very important, although growers do taste the flavor of fresh market carrots before they sell them, because flavor is very important to consumers and adds to the economic value of the crop. Some other traits we look at when we're selecting carrots, some of the selection is done visually, and we will continue to do this. A lot of the selection in the 1950s selected against yellow cores, shown in a couple of these pictures, and this has resulted in carrots with higher nutritional value when you select for orange cores instead of yellow cores, as Dr. Cholin is doing some similar work in this, I understand, in India. So how do we know what? So I've talked to you about traits like this, which we can screen very readily visually. What about more difficult traits that that can't be evaluated so readily? To address this question of first of all, identifying traits important for growers and consumers, we we put out a survey to about 50 to 75 carrot growers, carrot seed companies, carrot processors, and a consumer group to get some input as to what are the important traits that could be improved in a breeding program for carrots. And with this, we identified, we had a survey and ranked the response. And from a production standpoint, stand establishment or early germinating carrot seeds is particularly important to speed up the growth of the crop because it's a relatively slow crop to be established. Bolting tolerance or non-flower production for the U.S. crop is very important because growers do not like to see flowering carrots in their root production field. And then drought stress is recognized as being much more important because most of the U.S. carrots, the biggest production is in California, where drought is a persistent and increasing problem. We also other traits were on the survey, like heat stress, but they weren't in the top 3. Top pests that were suggested to be evaluated from a breeding and genetic standpoint were nematode resistance, cavity spot or pythium resistance, and alternaria leaf blight. Xanthomonas, not quite so important. And then from a consumer standpoint, sweet flavor was identified as a trait of importance to improve carrots for consumer quality, as well as new colors and more nutritionally improved carrots for the carotenoid or orange and yellow and red pigments, and for purple anthocyanin pigments. So these were some of the traits that we identified. The next step then was to put a team together, which Dr. Cholin mentioned. We've had some fairly nice grants looking at this, with about 8 collaborating scientists across the U.S. Asking the question, what diversity can we find in this collection of 700 cultivated carrots in the USDA collection for these 9 or 10 traits shown on this list? We can do some of the breeding in my program by myself, but we cannot do all this alone. Certainly plant breeding is a team effort, and this was how we started developing the team, by identifying traits important for growers and consumers. And again, growers want hybrids for uniformity, and that brings a lot of the uniformity to the crop, some disease resistance, early seedling vigor, which translates to top size. An early seedling growth translates to a bigger top, better resistance to flowering, shown here in the lower right picture, and lower input for organic systems, as well as abiotic stress tolerance. From a consumer standpoint, there's quite a bit of variation for carrots for things like sweetness and flavor and succulence, as well as for color. And so we identified new sources of both these grower traits and consumer traits in this large collaborative project that we've had. As we've moved forward on this project, we've put out some informational sheets for consumers, because while most consumers are familiar with orange carrots, we're seeing more and more purple, red, and yellow carrots, and some white carrots in the market for fresh market carrots in the U.S., and consumers are interested in them. So these are all carrots in the breeding program. So
Somebody's, can you please unmute? I think somebody has answered.
So this broad range of genetic diversity is what we're using to improve the crop. And I'll talk about the improvement process for 2 traits, one grower trait, one consumer trait that we focused on. For the grower trait, we've looked at nematode resistance. And this started in the 1990s when I was have been collaborating with Phil Roberts at University of California, Riverside, where we have identified a single dominant gene that controls nematode resistance. These are all carrots of the cultivar Brasilia shown in this picture. They're all grown in the same field, infested with nematodes. And I'm not a nematologist, but I can tell you which carrots are resistant. And so this has been a relatively easy crop to phenotype. In addition to phenotyping in the field, we also phenotype in the greenhouse to get accurate phenotyping. This is Melodigyne javanica we initially focused on. Incognita, Melodigyne incognita is a more widespread nematode that we've also focused on. As we've done breeding work, we've developed new inbreds. You see some carrot inbreds to the right there, fairly small like inbreds are, but big enough to produce a good seed crop. And in this case, very good nematode resistance, showing some of the genetic the pictures of some of the sources of genetic resistance that we found. I mentioned nematode resistance showing up in a Brazilian carrot. We also found nematode resistance showing up in Syrian carrots and Chinese carrots, in European carrots, and in Australian carrots in our earlier studies. So all these carrots to the right were grown on nematode-infested fields and came out very clean. When we looked at the genetics of the resistance, it's a single gene for javanica resistance, at least 5 genes for incognita resistance for those of you plant breeders. And as a plant geneticist, I'm required to show a picture of some chromosomes. And yes, we've put these genes on chromosomes, and we're in the process of developing good molecular tools to track these genes in a breeding program. So just to summarize for the nematode resistance work, started with evaluation for one nematode species resistance, Melodigyne javanica, moved on to incognita. Also major nematode pests for many other horticultural crops with several sources of diverse germplasm. We've released this germplasm and it's available to anybody that wants some seed. And I will note that with my big collaborative project we've identified new sources of resistance in this collection of 700 open pollinated carrots. One important source from South Africa, and also a very good source of Melodigyne incognita resistance for a plant introduction in the USDA collection from India. I'm glad to share what that plant introduction is with anybody that's interested in, if you want to find out the Indian source of nematode resistance. But we have this and other sources that we are working on to identify the genes controlling the trait. So that's where we're at in the process of nematode resistance breeding. Let's talk about a trait important for consumers, nutritional value. Nutritional value is an interesting trait because consumers can't actually evaluate nutritional value or select nutritional value as consumers, but they're very interested in it. Also, nutritional value has no dollar value to growers. A more nutritious carrot doesn't get a grower a higher value than a less nutritious carrot. So as part of the breeding process, we look at improving nutritional quality as we're improving other traits. Let's talk about the orange and yellow and red pigments that show up in carrot roots first. Actually, it's what we'll be talking about primarily. These are carotenoids or carotenes. These carotenoids occur in all green leaves. Leaves are green because of chlorophyll, but that masks these orange pigments that occur in all green leaves. And plants have to have the orange pigments because it's necessary to protect the process of photosynthesis from some of the byproducts of it. So that's why plants have carotenes in their leaves. And in addition to finding these pigments in leaves, you find it in the fruits of quite a few vegetables, for instance, tomatoes and peppers shown here. These are other carotenoid pigments, several of the 600 or so pigments. And some of these pigments are precursors to vitamin A. I'm not going to go into a long story about vitamin A, but lycopene is the red pigment of red carrots, and about also watermelon and tomatoes. Toward the bottom of the slide is beta carotene, which is the orange pigment in carrots and orange melons and sweet potatoes. And when we eat beta carotene, that molecule is cleaved in half to give 2 molecules of retinol on the bottom of this slide. And retinol is also known as vitamin A. So in fact, all vitamin A that is required for all animals comes from plant sources. It's not synthesized by animals. It comes from food that animals eat. So these orange pigments, beta carotene, alpha carotene, are very important. Why do we need vitamin A? It's an essential nutrient. It's required for human health. There are parts of the U.S. that are suboptimal in consuming vitamin A, not any vitamin A deficiency in its most extreme forms, but in much of the developing world, there is quite a bit of vitamin A deficiency yet today. After protein energy, major staple crop deficiencies, vitamin A deficiency is one of the major micronutrient deficiencies, with an estimated quarter of a billion kids every year deficient for vitamin A. And this results in those children and also often pregnant women having reduced immune function, meaning lower disease resistance, and in some cases, permanent blindness due to vitamin A deficiency. And maybe 500,000, primarily kids, die every year globally from vitamin A deficiency. So it's an important nutrient, important for US and important for the world. And so in order to improve vitamin A, you start with carrots that have more of these orange pigments. You can see some of the range of vitamin A content just by looking at these carrots. If you cut them open, you can look better, but the orange carrots here with the darker orange color, in fact, have higher vitamin A content. And so we've selected for vitamin A content visually, but also using chromatography to select for higher pigment content. It turns out that the control of the color of carrots is relatively simple. There's about 3 major genes that control the colors, the difference between white carrots, wild carrots shown on the left here, but also white cultivated carrots also have the same genetic control. Yellow carrots is 1 gene different, shown to the upper right here, from white carrots, and in orange carrots is 2 more genes different from yellow carrots, shown in the lower right. We're working on the genetics of red carrots, and that's 2 more genes beyond the 5 genes that I mentioned already. In addition to these genes that control the overall color of carrots, there are other genes that control the quantity of color, the darkness of color, and about 20 of them control dark pigment color. So we have orange carrots that have as low as 50 parts per million. That number doesn't mean anything to you until I show you the next slide. Orange carrots can have 50 parts per million alpha and beta carotene up to 1,000 parts per million, controlled by these 20 genes that we are mapping and tracking. And we've released some dark orange carrots in the breeding program. And the release of these darker orange carrots, as we've improved other traits for carrots from the USDA breeding program, has had a positive impact on the US carrot crop, because in the 1970s, when my predecessors were breeding carrots, and right before I started working on carrots, the average carrot in the U.S. had about 90 parts per million carotenoid. I'll tell you what 90 parts per million means in the next couple slides, but it gives you an orange carrot with a relatively pale color, often with a yellow core. By the 2010s, selection for darker color and only orange cores, no yellow cores, has resulted in all new cultivars having about 130 parts per million, about 50% higher content of these vitamin A precursor compounds in carrots sold in the US today relative to carrots sold in the 1970s. What does this look like? Well, starting with a breeding population shown with slices in the middle, with about 140 parts per million carotenoids in those carrots. We selected a couple of populations with 2 times that amount of carotenoids on the left, or 3 times that amount of carotenoids on the right in the breeding program in the last couple decades. And so, as I mentioned before, it's a matter of intercrossing dark orange carrots to get darker orange carrots. And over the years, this has led to quite dark orange carrots being sold typically across the US. As I mentioned before, though, consumers can't really select much for color because they have to buy what's on the in the market. The other thing is but consumers do recognize carrots are nutritious. But what consumers are really interested in is flavor for fresh market carrot sales. And so as we've improved nutritional quality, we've paid a lot of attention to flavor with grad students I've had like Doug Sinalik, because in order to improve consumer quality, flavor is paramount. It's the top trait of importance. So if you look at average carrots, the average carrot sold in the U.S. in 1975 versus to 2015, or actually yet today, about 90 parts per million carotene content, up to 130 today. Consumers are eating more carrots thanks to the convenience of baby carrots. So the per capita U.S. consumption is up. And because of this, the contribution of carrots alone, even with only 5 kilograms per capita per year eaten by U.S. consumers, carrots contribute about 12 to now about 14% of the vitamin A in the U.S. diet from these orange pigments in orange carrots. Carrots is one example, but other crops that you may be working on might be bananas, shown in this image to the upper right. These are different. This is some diverse germplasm of bananas, but also melons, papaya, mango, sweet potatoes, all vary for carotene content and can be very good sources of vitamin A. So we've done quite a bit of work on carrots, but there's lots of work that can be done to improve nutritional content in breeding programs for other crops. But as you're doing that, you need to think about what the growers also need and what the consumers are interested in. So this is where we've come so far with carrots in the breeding program. Some examples of some things we've been doing. A question I get is, why are we doing carrot improvement? Who needs improved carrots? Carrots are in the market every day. Growers have seed available. Who needs better carrots? I hope that I've convinced you that growers can benefit from improved carrots. With these nematode-resistant carrots, the growers will be able to reduce or probably eliminate the use of a pesticide that's damaging our ozone layer. And so that's something that carrots are being improved for, that our program is targeting, so that we can improve the crop for growers to reduce the cost of growing carrots economically and the environmental impact of carrots with these pesticide resistant carrots. From a consumer standpoint, we would like consumers to eat more carrots, as carrots being one of those vegetables that most Americans are not eating enough of. And so from a human health standpoint, consumption of more carrots and other horticultural crops certainly is of great value to the consuming community of consumers around the world. So I've given you a brief overview of the history of carrot crop, a little bit of a history of the development of hybrid carrots in the 1960s, baby carrots, these short pieces of carrots developed in the 1980s. I talked a little bit about some of the breeding tools we've developed for accelerating the breeding process, some of the importance of breeding for nutritional value along with flavor. And talked about some of the diseases that we have resistance for. There are certainly quite a few other diseases we do find resistance for in carrots. We just haven't worked as extensively on them. So that carrots today really are a high value crop. I didn't mention it before, but those baby carrots, if you look at per kilogram of carrots, we in the grocery stores are paying about 3 times as much per kilogram of baby carrots as for whole carrots. So consumers value that convenience, and it has increased carrot consumption. So this has been important to make a higher value carrot crop for our growers. We're looking at developing carrots for lower input, which has economic positive impact, but also environmental impact. And we see an increasing amount of organic carrot production in the U.S. It'll probably continue to rise. And what we paid a lot more attention to in the last 5 to 8 years is abiotic stress. We hadn't been looking much at abiotic stress earlier in my career, but this has become very obvious that we need to have carrots that are able to grow in saline soils in warmer, drier parts of the world. And there is substantial genetic diversity for these abiotic traits as well. I didn't show you images of that, but it's very striking both in cultivated and in wild carrots. So this is a picture of a farm family in Wisconsin where I'm living from the 1850s. This is a small team of horticulturists growing some vegetable crops shown here, as well as some corn, of course, and carrot improvement, like any crop improvement, is a team effort. Alone, I can't get very much, maybe nothing done, but with a team, we're getting quite a bit done. Thanks to the ability to be training graduate students, focusing on carrots. I also worked on a few other crops Dr. Cholin mentioned. Also with very capable help from Techie Doug Sinalik, and funding. My basic funding is from USDA-ARS. That's where my salary comes from, but other funding sources along the way where we've competed successfully for competitive funds in the U.S. So carrot improvement is a team effort. I welcome anybody to join the team. We're a small group. We can always use more people. And I would invite any questions you might have with my presentation.
Thank you so much, sir, for your elaborative discussion and sharing your experience, starting from germplasm expedition, domestication, genomic selection, and many more. The breeding strategies in terms of need of carrot for growers and consumers is a great lesson to a public sector scientist like us. So I'm sure the students and scientists have been enlightened with your presentation. So I would like to first introduce our Honorable Vice Chancellor, Dr. K. Indiresh, sir. He would like to talk to you. Sir.
Yeah.
Sir, thank you, sir.
Am I audible?
Yes, sir, you are audible, sir.
Yes.
Thank you, Professor Philipp, for giving a very good presentation on the carrot breeding, advances in carrot breeding.
Thank you.
In fact, in India, we are mainly concentrating on tropical carrots rather than temperate types. And most of these tropical carrots are known for high carotene content and also vitamin A. In fact, these orange colored carrots are dominant over the other colors. And in terms of yield also, we are getting good yield in some of the climatic zones in India. And a lot of nowadays, the breeders are working on biofortification of some of the vegetables, where we can enrich the carrot with vitamin A and other nutrients. Is there any work is being carried out on the development of biofortified carrot types or carrot hybrids? And since you are working on carrots, are you working on the biofortification in case of carrots? This is my question to you, sir.
Yes. The carotene improvement would be an example of biofortification. We're also looking at other things in carrots, like mineral content, where something like iron or zinc and magnesium would be positive, and calcium would be positive minerals that accumulate in carrots. And then there's some minerals like cadmium and arsenic, which have a negative nutritional value, that also are variable in different cultivars of carrots. So this would be some other research we have underway relating to biofortification. Also looking at variation in the purple pigments. I showed it briefly. The purple pigments are very good antioxidants and probably involved in a fair amount of disease avoidance or diminution of disease disease patterns for humans. These purple pigments, and so these are some other nutrients we've looked at. I'm talking with Dr. Cholin. We've got she has some great ideas for some other micronutrients to improve on carrots. So yes, the list is quite long, and there's lots more that can be done. But thank you for your overview of your Indian breeding effort, I do realize India is a is a country with major carrot improvement programs. So.
Thank you, sir. I would like to introduce. Thank you, sir. I would like to introduce Director of Research, University of Horticulture Sciences, Bagalkot, Professor Maheshwarappa, sir. Sir, please.
Hello.
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 Sir, you are audible, sir.
Sir, sorry.
Yeah, you are audible, sir. Please.
Sorry. Thank you, Dr. Philipp, for the informative talk on the carrot breeding. Actually, we thank on behalf of our university for the very good information. And I think your help in transferring or exchange of the germplasms through to our university, it will have a very good impact, or will be improving our breeding work in the times to come. Thank you very much for the informative talk. Definitely, our Dr. Saramala Cholin will carry over the work on breeding aspect. The germplasm which you have given to us will have a good impact on developing new hybrids in our country. Thank you very much. Thank you, Saramala.
Thank you, sir. Sir, there is one question. There is
Yeah.
one question from Dr. Harish. Excellent session with the overall perspective. Can you show some more light on breeding low input responsive carrot types, especially for warmer climates?
Yes. So for low input, well, some examples of low input would be disease and pest resistance. At this point, we have done a small amount of work looking at lower fertility amendments for carrots. Carrot can be grown with a very wide range of fertility sources, ranging from nitrogen applied by conventional growers to many sources of animal waste used for as nitrogen sources in organically grown carrots. In terms of reduced input, to be very honest, we haven't had that as a major breeding focus. We do know that there's a range of variation when we've looked at, say, reduced nitrogen for the crop, but it's a trait that, based on our very small preliminary work, is open for improvement. So if you're interested, I would say pursue it, but unfortunately, I can't give you any more detailed information on it. So that it's a trait where we haven't. I guess I wonder what other, when you say low input, what other lower input? We are looking at lower water input as well with drought tolerance. So that's another example where we are doing some work, and there's a wide range of variation. But I'm not sure if I did answer your question about the whole range of low input, though.
Thank you, sir. So one more question, from Manoj Sutradhar. Very informative session. What are the qualities you are targeting for future breeding programs?
Well, as we go along, we don't reduce the traits we're looking at. We tend to continue to look at the traits we've already been working on. To some extent, something like the development of male sterility is well established, and we are doing a little bit of work for improving that as we move forward, but not so much because we have a good system in place. But as we move forward, I would say abiotic stress for sure is the most important trait. There's going to be more and more human population to feed. There's going to be more challenging production due to climatic conditions around the world. And in terms of the future, I would say that has to be an important collection of traits that we focus on, because we have to continue to provide consumers with carrots and other vegetables.
Thank you. So there is one more question from Dr. Prabhudev Vajjapalawar. Are there any agronomic practices which influence the carotene content in carrot?
Good question. Not much. The carotene content is, the way I would put it is a carrot that is growing well. If the grower is looking at the crop and it's growing well, it's generally going to have an average amount of carotene. There is relatively minor reduction in carotene content under drought and under some altered fertility practices. It's interesting that carotene content is relatively unaffected. Certainly, certain growing areas will have overall a little higher or a little lower carotene content. That can be a function of the field conditions, but also the time that the crop is in the field. In other words, the developmental, how long has that crop been in the field? Because carrot continues to get some accumulation of pigments later in the season, not as steep, but it continues to accumulate pigments to some extent as the season goes along. So an early harvested carrot would have less carotene, but overall, the ranking of content of carotene in different growing conditions is almost never affected much.
Thank you. Sir, one more from Dr. Shankarappa, who is a pathologist. Any work on powdery mildew and alternaria resistance?
Yes, powdery mildew is a trait that did show up on the survey. I didn't show it to you. It was number 5 after Xanthomonas, and it is an important trait. It seems to be under relatively simple genetic control. That's where things can go. In terms of work we've done, we haven't done much with it. We take notes on it when we see it. We don't see it regularly, and so that's one reason. If you're doing any disease resistance work, you need to reliably see the disease attack. And we don't see powdery mildew showing up regularly. When we do, we take notes, so we know there's variation. It is a bit of a growing concern to the growers, they tell me, but it's primarily because of new cultivars that are less powdery mildew resistant. But I think it seems like it's under relatively simple genetic control. It's something we are not working on, though. So if you want to work on it, I'm sure there's a project there for you to pursue with some relatively simple genetics. Alternaria, on the other hand, is a much more complicated trait, genetically speaking, and we do have a fairly large effort on that. Wild carrots actually are a better source of alternaria resistance than cultivated, but there's some very good cultivated sources of resistance. It's a genetically complicated disease, and we do have some relatively alternaria resistance carrots. I'm providing some seed to Dr. Cholin to an inbred called 5280, which 5280 is quite good alternaria resistance. And so we are focusing on that. And at this point, I'll say it's going to be a long process to have uniformly resistant. There is no such thing as completely resistance to alternaria leaf blight that we've found. But we do find a wide range of tolerance.
So, a couple of questions from Dr. Rajesh. Why the importance is given on improvement of purple color over orange color? What about acceptability of purple colored carrots at consumer level?
Yes, yeah. Purple carrots are interesting. For U.S. consumers, some people won't put a purple carrot in their mouth because that's not the right color for carrots. So but kids will eat purple or yellow or red or carrots of any color. So what we're looking at is improved flavor in particular. The color, the purple pigments themselves have no negative effect on flavor, but the genetic sources of the genes for purple that we've used, primarily coming from Turkey, China, and a little bit in India, those genes for purple color are coming from carrots that do not taste very good as fresh market carrots. A lot of the purple carrots that apparently have been consumed historically were processed, and so consequently the flavor that we perceive as a raw carrot, which is my focus, is fresh market carrots. So we're looking at moving those purple carrot those genes for purple color into carrots that are well adapted for the U.S., but also with a big focus on flavor, because the flavor is not very good typically from the gene sources we have. Again, that's not caused by the purple color, but just comes along with those genetic sources. Another challenge we're seeing with purple carrots that from your perspective maybe isn't a challenge is that we see a lot of early flowering coming from these sources of genes for purple color. Again, these purple color genes don't control flowering, but they come along with that whole package of genes from those carrots. And so it's bringing a focus to selecting for control of flowering as we're incorporating purple into carrot cultivars.
Thank you, sir. So one more question from Chandan. What do you think is the major hurdle in the molecular breeding program of carrot? He's working on
the major challenge is getting molecular tools that are inexpensive and fast to use. I think that we have the capability of doing a lot. That's from the tool side of it. From the breeding side of it, it's getting good markers for all the traits that we have. Those are, that answer I think is pretty comparable to any other crop, is that we need more markers and we need tools that can be used more quickly. I don't think it's any different for carrot. The fact that there's relatively few of us working on carrots, means that the development of the tools is slower than it is for crops where there's many researchers. But I'm confident Dr. Cholin and others in India will be coming in to help develop new molecular tools to improve the breeding process.
Yes, sir. So one more question from Dr. Mahantesh Naik, who is a molecular biologist. So his question is like, any attempts on genes associated with pathways linked with quality traits?
That's a great question. The question is, what are those genes? So we initially thought, gee, for carotenoid content, these must be carotenoid biosynthetic genes. So we started focusing on biosynthetic genes in the carotenoid pathway, thinking that that's where we would find the genetic regions that control carotenoid content. That's not the case. The carotenoid color genes are accumulation genes, not biosynthesis genes. So these dark orange carrots and orange carrots in general are a better sink, a better storage vehicle for carotenoid pigments. One thing I'll point out is, why do carrots have this range of colors shown in this picture? It's only because of humans. Wild carrots don't have these pigments. So as humans, as our ancestors selected for carrots, for colors in carrots, they selected this range of colors. So something like carotenoids have no function in the carrot root to the carrot. And so that may explain why biosynthetic pathways aren't the reason, or aren't the basic variables for much of the variation for those pigments. But at any rate, I would say we look at biochemical pathways as a byproduct of other things we do. When we're looking at QTL analysis, at individual gene analysis in a segregating genetic population, we will ask the question, where is the gene, of course, and then what might be the candidate gene for that trait. Sometimes it is a biosynthetic gene, but if it's not, we'll go with whatever the carrot's doing and try to better understand what the genetic mechanism is for the molecular process of accumulating these compounds.
Thank you. So one question from Impas student. What are the climatic and soil requirements for growing the baby carrots? So we don't grow baby carrots in India.
Right. Well, the climatic conditions are the same as any other carrots. The soil conditions are very important, because though we can get carrots up to 40 centimeters long, but that means you have to have a farm equipment that's breaking the soil up that deep. If the soil's only broken up 10 centimeters, the carrot that has the genetic potential to be 40 centimeters long will only be 10 centimeters long if it hits a hard pan of soil. So that it turns out that the soils of California are fairly sandy for most of the carrot production regions, and so it's quite good for growing long, thin carrots. So if you're thinking of growing baby carrots, those long, thin ones cut into pieces, get some good farm equipment people to help to work with you.
So one more question: astringency and carotene have any correlation? If so, what are the approaches for reducing astringency without compromising the quality standard?
Yeah, that's a good question, because I think what you're referring to as astringency, we refer to as harsh flavor or turpentine flavor. And interestingly, those chemicals that give carrot most of their good and bad flavor are 10 and 15 carbon volatile terpenoids. Carotenoids are also terpenoids. They're tetraterpenoids, and so they have 40 carbons. So you might think there could be some relationship between carotenoid accumulation and carotenoid biosynthesis and flavor. In fact, there's none to speak of. Or, I mean, if there's any relationship, it's because of linked genes, not because of cause-effect of those genes. So yeah, luckily, we can breed for each of these traits independently, as long as the genes aren't too closely linked in the breeding program, but that's where the molecular tools will help.
One more question. So can you please throw some light on the DH line production in carrot, double haploid lines?
Yeah, that's a great question. So DHs are doubled haploids. Almost all of them, actually all of them to date, come from anthroculture. There are a couple of the seed companies are focusing on this, but anthroculture works for some carrots, but there's a strong genetic component to anthroculture success, and many carrots just don't respond positively to haploid production in anthroculture. So to solve that problem, there's a couple of projects globally, one here at Wisconsin, looking at using mutations of histone proteins of the centromere, CENH3, as a way to universally form haploid carrots. It's looking promising, but so far the technique has not been fully developed. I'll also mention there's some work ongoing with editing genes here, and that works pretty well. That'll be a great tool for looking at future tools for carrot and other crop breeders, genome editing, if it's allowed. And so far it is going to definitely be a tool in the toolbox of all future plant breeding programs.
One more question, like what are the practices you follow for protecting the populations in the process of inbreed development?
Of inbred development?
What are the process? Yes, while having the inbreed, while doing inbreed development, how do you protect the populations, allelic richness and all? So allelic diversity, how do you maintain that?
Well, so when we're selecting inbreds, we will start with several populations that we eventually look of sibling or very related populations from the same cross that we are interested in developing inbred from, and we will carry along 4, 5, 6 different populations from that same cross for as many generations as we can, with the idea that we want one of these populations to be the source of a new inbred. We don't really want more than one, but we don't know which is the best population to start with at the F3 level. So we'll carry several populations along for each of them. That's the best way to make sure that you're going to get an inbred from a given cross.
Thank you. Sir, one more question from Dr. Suma Mugli. Carrots with anthocyanin are drought tolerant and also insect tolerant.
We haven't noticed that. Maybe so. I wouldn't dispute it because we haven't tested that extensively. We did have some purple carrots in some drought tolerance work. I can't say we saw a particularly great drought tolerance. Not that it was bad, but we haven't tested as much purple germplasm. So you may well be correct. If it's true, I can't explain why. I wouldn't have a good idea. But I'm interested to hear that. Now I'll look closer at our data, see if we maybe found the same thing but hadn't recognized it before. So thank you for mentioning that.
Thank you. One more question from Chaitra Kulkarni. She has worked with me on Asiatic and Western carrots. So have you compared the flowering behavior of Asiatic and Western carrots?
Yes, we have. And Asian carrots flower early. and too early for Western European and U.S. production, because the carrots grown in Western Europe and the U.S. all came from the carrot breeding efforts started in Europe, which had a heavy focus on required vernalization, which is not a natural requirement of carrots. Your carrots in Asia, your local carrots that don't come from European sources, I don't think have hardly any selection for vernalization, not to the extent that European carrots. So yes, when we grow carrots, the landrace or local cultivars of carrots from India or any of the warm regions of the world, we get significant amounts of early flowering, and that's why we're tracking the genes controlling vernalization.
One more question, from Dr. Ambika. Can you share some information on management of soft rot disease? That is a major issue in our place.
Yeah, I think this is probably used to be called Erwinia. I think it probably has I think it has a new name, but again, management. The best I recommend. So we see Erwinia rarely, and it's almost always after something like a flood, which leads to anaerobic conditions in the soil. Obviously, you can have these pathogens in soil that are not in anaerobic conditions. So from a management standpoint, avoiding those anaerobic conditions, at least from what our experience in the U.S. is, and I think it's the same in Europe, avoidance of anaerobic conditions in your field, which typically come from things like flooding, would be the best way to avoid it. Of course, it's very difficult to avoid flooding. Again, there is a genetic component, so that's another approach to it, is a breeding approach. That's a longer one. But from a management standpoint, I don't have a lot of experience, but I've shared what I do know, though.
One more question, maybe last question, I think, is left with few minutes. So the question is, what are the efforts made to understand the role of microRNA, which may be involved as decoys for carotenoid biosynthetic pathway gene?
Yeah.
So leading to the difference in gene expression.
Yeah, that question is wide open. We have some evaluation of that, but we're very early in looking at microRNAs and some of the other things like some epigenetic variations as well. There's a lot of room for research in that area. So if you're doing any work like that, I'm glad to collaborate, because we are not doing too much now, and we'll be glad to see what you're learning, because lots yet to be learned about that.
Yeah, one more question from Santosh, who is my student, and he's with Professor Darius in Poland now. So what are your thoughts on transposable elements that affects on difference in vernalization requirement in temperature?
Yeah. No, my thoughts is I'm glad you're working with Darek Jebelus, because he started working on transposable elements in my lab when we discovered a trait conditioned by a big insert. Yeah, you're working in the best place in the world with transposable elements in carrots. And lots of interesting things with transposable elements. They're certainly the cause of quite a few traits. We see large inserts or deletions, primarily inserts from transposable elements that go along with alleles for quite a few genes. Be nice to know if we can somehow control them from a breeding standpoint with something like ACDS type of a system. Yeah, keep studying it, because what you're learning will be very beneficial, not only to help us understand the carrots we have today, but where we might be able to use transposable elements to better somehow breed carrots. It's a very interesting area.
Thank you, sir. Thank you very much.
Thank you. So thank you, Professor Philipp Simon, for taking a lot of time on presentation and clarifying the doubts. So I'm very fortunate to be associated with you, and I thank University of Horticulture Sciences Bagalkot NAHP-IDP for making this visit possible for me. Thank you so much, sir. So.
Thank you for listening, and I really appreciate the opportunity to talk to you and to have some conversations, and hopefully can collaborate with not only Dr. Cholan but others of you as well. Thank you.
Thank you. Thank you, sir. Thank you.